Transporters are an interesting type of spanning protein because they conduct against concentration gradient like pumps, but they are not using energy. That is unlike pumps. They don't have ATPase activity, so they are conducting substances against concentration gradient without using energy. They change their conformation while conducting, and they select substances on the basis of their sizes and shapes. We'll see how they conduct when they are not using energy and against concentration gradient in the coming slides. The transporters, they could be uniporter, or they could be multiporter or co-transporter. Uniporter mean if they are conducting only one substance in any direction into the cell or outside of the cell, they are called as uniporter, conducting one substance. Like glued channel glucose transporters. Glucose transporter are called as glued. These glucose transporter, they conduct glucose against concentration gradient from lower concentration to higher concentration. They are present in small intestine for absorption of glucose, in the kidneys, in nephrons for the reabsorption of glucose from filtrate. And then multiporter and co-transporter. Multiporter or co-transporter are the transporter that conduct two substances or more than two substances in either same direction or opposite direction. If they are conducting in opposite direction, then they are called as antiporter. For example, a transporter that is conducting calcium into the cell and potassium, sorry, it is sodium outside of the cell and calcium into the cell, or chloride and bicarbonates in opposite direction. Symporter are the transporter that are conducting sodium and glucose, for example, in same direction. They are conducting sodium into the cell, and then glucose is going into the cell in the same direction. They use the potential gradient of one substance to conduct another substance. For example, here the sodium is more outside of the cell in case of intestine anywhere. So glucose, if it is less outside of the cell, when sodium goes into the cell along its concentration gradient, it will take glucose along with it. So they are called as symporter because they are conducting sodium and glucose in same direction. Here we can see a uniporter that is conducting a substance into the cell. These protein, transporter protein, they are also called as carrier protein. So they bind with a substance and change their shape to send these substances in to the cell or outside of the cell. So here we can see a symporter that is conducting glucose and sodium in same direction into the cell, from outside of the cell into the cell. uh, that is called as a symporter. Along they are conducting sodium along its concentration gradient, and by using the concentration of sodium, they are also conducting glucose against its concentration gradient. We can see sodium is more outside, it should go into the cell because its concentration gradient is toward inside, but glucose is more inside, it should not go into the cell if there is no symporter. So this symporter, it is using concentration of sodium, it is passing sodium along its concentration gradient, and glucose is also taken into the uh, cell by using concentration gradient of sodium. When sodium get bind to this transporter, it changes its shape so that the glucose get attached to its domain, and then the sodium is passed into the cell, and along with that the glucose is passed against its concentration gradient into the cell. Most of the antiporter and the symporter, they are linked with a pump, so they are not using energy directly but indirectly. So this pump here, this symporter sodium glucose symporter is linked with the sodium potassium pump in order to keep the concentration of sodium more outside For example, if this transporter keeps on conducting sodium into the cell, after time, if there is no pump, sodium would become more inside the cell as compared to outside of the cell. So in order to keep the concentration of sodium maintained outside, there is a pump. It is conducting sodium outside by using energy and potassium into the cell. So sodium concentration is always more outside of the cell, so that the transporter or symporter can take the sodium into the cell along with glucose against its concentration gradient. So here we can conclude transport across cell membrane into the cell or outside of the cell. We know there are mainly two types of transport, that is the uh, passive transport, and then there is active transport. The passive transport is the transport without using energy, and active transport is by using energy. So passive transport is in the form of diffusion and in the form of facilitated diffusion. The diffusion is occurring across cell membrane directly, and we know the diffusion is passage of substances from higher concentration gradient concentration to lower concentration, that is a long concentration gradient. So then the facilitated diffusion, it is through channels. For example, we know the substances, they are being conducted through open channels or closed channel along their concentration gradient. And then there is active transport. Active transport could be primary or it could be secondary. The primary active transport is through pumps, because the pumps, they are using energy directly, so it is called as primary active transport. And it is active transport because it is by hydrolyzing ATPs. And secondary active transport is transport done by transporters, that is by uniporter, by symporter or antiporter. They are using energy indirectly because they are mostly linked with pumps to keep the concentration of one substance higher so that they can conduct another substance. So they are not using energy directly, but they are using energy through the pumps that are linked with them. So that transport is called as active transport. Then another transport is called as cytosis. Cytosis means the transport through the cell membrane by the by engulfing, by making the uh, bulges into the cell or outside of the cell, that is called as exo. When the cell membrane bulges out, it is called endo. When endocytosis, when the cell membrane bulges in, and when there is transport between cell, then it is called as transcytosis, one cell to another cell. So cytosis means breakage of the membrane to pass material, and it is for the larger materials that cannot pass through diffusion, facilitated diffusion, or primary or secondary active transport. Cells release their larger products in the form of vesicle outside of the cell through process of exocytosis. We know these cells, they have genetic material, DNA, and these DNA, they are transcribed into the messenger RNA. These messenger RNA, they go out of the nucleus into the cytoplasm, where they are translated on ribosomes into proteins. Then these proteins, they may enter into endoplasmic reticulum, and from endoplasmic reticulum processing occur, and then they enter into Golgi apparatus. Golgi apparatus are sac-like structures. So these sacs, they process and package the proteins. So these proteins, they come out, bud off from the Golgi apparatus in the form of vesicle. Because Golgi apparatus are made up of plasma membrane, so this plasma membrane is present in these vesicles. So these vesicles are made for the exocytosis as well as for use inside of the cell. So how these vesicles decide either they have to go out or remain inside is depend on the marker proteins. So there are marker proteins present on the vesicles. They are same like we studied in the plasma membrane. These protein they are called as vesicular snare protein. So then there is a target snare protein on plasma membrane. So these T snare and V snare. Vesicular snare is V snare and target snare is T snare. These two snare protein they can recognize another protein. That protein is present in cytoplasm. This protein is actually linker. It can link. Here we can see the green protein. It can link the T snare with the V snare. So when this protein can link these two protein when there is calcium. So calcium comes into the cell from the by using calcium channel, and these calcium they come in between because these protein they are negatively charged, so calcium attack these protein together. So when these V snare and T snare they attack to the snap, the green one, then they get fused to each other, and then the membranes, the vesicular membrane and target membrane, they get fused, and then material is released outside. That is called as exocytosis. Here we saw the V snare, vesicular snare protein, marker protein has recognized the target snare protein through a snap protein by using calcium. Why calcium is important? Because calcium is positive ion and these proteins they are negatively charged. Calcium comes in between and calcium may bond with them to attack them. In endocytosis, the substances are brought into the cell. The material to be internalized are surrounded by an area of cell membrane which then buds off inside the cell to form a vesicle. We know there are two types of endocytosis, that is the pinocytosis, that is cell drinking, and phagocytosis, cell eating. The endocytosis could be receptor mediated or also called as Clattering dependent endocytosis. The Clattering mediated or Clattering dependent endocytosis is done by the production of small vesicles that have morphological characteristic coat made up of cytosolic protein Clattering. So the receptors that are attached to membrane, when they get linked to any substance that have to be taken into the cell, these receptors when they get bind to substances they are then bound by a protein that is called as Clathrin. Before attachment of the substance, these Clathrin protein they were uh, in soluble form, and when these substances they bound to the receptor, these Clathrin protein they precipitated out and they have enclosed these receptors. So it is called as Clathrin coated uh, vesicles that are formed, and then these vesicles when they are internalized, then these Clathrin protein they get detached from these receptors, and then the this vesicle it get attached to the lysosome to make the early endosome. Another type of endocytosis is done by cavuli. Cavuli are the receptors that are not coated by the uh, Clathrin, and they are the receptor that get attached to substances, and then vesicles are formed. These vesicles then are internalized, and then there are formation of endosomes. There could be another transport type that is called as protocytosis. That is by using cavuli vesicles to bring molecules of various sizes into the cell. Uh, these protocytosis, they do not send substances to lysosomes or other organelles. Uh, but in case of protocytosis, the substances, they are directly released into the cytosol, so there is no formation of endosomes or secondary endosomes. So protocytosis is done by receptors that are not coated by clathrin, and then these substances or these vesicles, they are not going into to fuse with lysosomes. They are releasing their substances directly into cytosols. Pinocytosis is occurring along the highly ruffled region of the plasma membrane, where the invagination of the plasma membrane are formed to form a pocket, and then these pockets are picked off to form vesicles, and then these vesicles, they get attached to lysosomes and form the endosomes. There could be phagocytosis, that is again non-clathrin mediated. The larger substances, they are directly taken up by the cells by making the uh, invaginations or vesicles, and then they go to the endosomes by the fusion and with the secondary endosome by fusion with the lysosomes. So the endocytosis is mainly of 3 or 4 types, that is mainly of 2 types, like the clathrin mediated and clathrin non-mediated. Clathrin mediated mean the clathrin protein get attached to the vesicles, and the clathrin non-mediated with the help of cavuli, and if cavuli get attached to each other make endosome, and then get attached to lysosome to make secondary endosome, then it is called as the transport done by cavuli. If this cavuli, they are these are the receptors. If they are not going to attach with lysosome, but they are releasing their substances directly into cytosol, then it is called as podocyte. And then the protocytosis, and if the cavuli, they are taking the uh, material, sorry, if the pockets of substances are directly made into membranes, and then these are taking the fluid material, then it is pinocytosis. And if the material are solid, larger material, they are directly taken up by the membrane without receptors by making only invagination, then they are called as phagocytosis. So phagocytosis and pinocytosis, they are not done by receptors. The podocytosis and cavuli and the uh, endocytosis, clathrin mediated endocytosis, they are done by receptors. For transcytosis, first we have to see the structures that are connecting cell to each other and to the surface where the cells are attached. So there are mainly 3 types of connection between cells and with their surfaces. These connections, they are either tight junctions, desmosomes, and hemidesmosomes. So the tight junctions, these are the connections. Tight mean they are very strict. They are between the membranes of 2 cells. They are stitching 2 membranes together. They do not allow passage of substances between the 2 membranes. They allow only passage of substances through cells, not in between 2 from the spaces between cell membranes of two cells. So they are called as tight junction. Tight junction are having two zones, a zonula occludens and zonula adherens. They are the zones, like zona occludens is a zone that is a stitch zone. It is made up of fibers that are stitching the two membranes, and then these fibers they are passing to other membranes of the other cells and stitching them. So they are continuous fibers of the uh, protein that are stitching membranes. So they are making tight junction, they are making a zone that is very adhered. And then there is a zona adherence. Zona adherence is made up of protein that are glue like protein. They are just attaching to membrane, but they are not very strict. So tight junction is made up of zona occludens and zona adherens. Zona adherens is again, this glue is made up of protein. So these protein they are attaching to membrane but not very tight like zona occludens. And then we have desmosomes. Desmosomes are the proteins that are fibers that are attaching to cell membranes, uh, and these protein they are contributed. These are the connection protein or junction protein. They are contributed by both cells. And in case of hemidesmosomes, the hemidesmosome is present between cell membrane and the surface where the cell is attached, because cell can produce protein. So these proteins they are only contributed by cells to attach cell with the non-living surface. So the this junction is called as hemi, mean half desmosome, because the fibers are only contributed by cell, not by the dead material that is here. It is basement membrane. So if it is contributed by both cells, then it is called as desmosome. When it is with the surface, it is only contributed by the cell, not by surface. So they are called as half desmosome, hemidesmosome. Then we have gap junction. Gap junction are actually channels, but they are present uh, between two cells. They are spanning the membranes of two cells. So they are for passage of material from one cell to another cell. They are actually helping in transcytosis. The substances they may pass from one cell to another. So the ions they may pass from one cell to another. The infectious material, viruses and bacteria and other, they may pass from one cell to another through these gap junctions. So there are three types of junctions: tight junctions, desmosomes and gap junctions.